the same number of chromosomes. With the same number of chromosomes. In SCB 204, you'll discover the other type of cell division that happens specifically only in one type of cell group will actually, the cell that divides will produce daughter cells, but it'll have lesser the number of chromosomes. Only 23 chromosomes, not 23 pairs. But don't write that down. That you'll learn about more specifically in SCB 204. But here in this course, we'll learn about mitosis. And mitosis is the type of cell division that will occur just about with all of your cells of your body. With all of the cells of the body, meaning your skin cells, your bone cells, your muscle cells, your gland cells, etc., etc. Every single cell of the body, with the exception of the reproductive cells. The reproductive cells, which are which? Which are the reproductive cells? In men, they are called the cells. The name, not the cell, not the name of the organ, but the name of the cells.
Sperm.
Sperm, good. And in females, the name of the reproductive cells are called eggs. So sperm and eggs do not divide by mitosis. Excuse me. They divide by the other type of cell division that you'll learn about in SCB 204. But every other cell of your body, with the exception of sperm and eggs, will divide by mitosis. Now, there is also one other exception. A group of cells that cannot divide because they don't have centrioles. Remember, centrioles are necessary for cells to divide. Centrioles, if you remember, we saw that last week. Well, I don't have it here, but maybe on one of these small models, you might have some representation
Bullets.
of centrioles. Well, I do see some representation of centrioles. Those two little things that look like little pieces of macaroni or pasta, look like two little things that every cell had a pair of. Well, remember, neurons do not have centrioles. So neurons cannot go through this process. Okay, neurons, by the way, do not divide at all. The neurons of your nervous system, of your brain, do not divide. But every other cell of your body, with the exception of neurons and the reproductive cells, will can divide by mitosis. Okay, so now, in your books, you'll see lots of pictures, and in diagrams and in PowerPoints, you'll see lots of pictures of something that's called the cell cycle. So let's look at the cell cycle. The cell cycle, right? In the cell cycle, you'll have different parts of the cell cycle, where one part says a G1, then there's G2, then there's S phase and the M phase. We're not going to go into all those different parts of the cell cycle, except for a couple of important things. First of all, during the cell cycle, we have a large part of the cell cycle, meaning throughout the cell's lifetime, where the cell is in one major part of its process called interphase. Okay? So a large part of the cell cycle is what we refer to as interphase. Okay? Now, what is interphase? Again, going back to the cell cycle, it's all the different events that happen throughout a cell's lifetime. Whether the cell has a very long lifetime, like some white blood cells. I'm sorry, you gotta hide that drink into your bag. Okay? Okay, and cells that have a very short lifetime. Cells that will mature into red blood cells live only for a few days, don't live very long. So it varies. However, every cell has a certain period there that is called interphase throughout its cycle. Now, interphase is not part of cell division yet. It's not part of mitosis. Okay, but it is a major part and a large part of its lifetime, where the cell is not divided, cell not divided. Okay, right? So first of all, if the cell is not dividing, if the cell is not doing this yet, it's within some tissue and it's not dividing, it's not multiplying, then what is the cell most likely doing then? No, not yet. If it's not reproducing, if it's not divided, then what do you think the cell might be doing?
Growing.
Getting its energy.
Can it be growing? Yes, it might be growing. What else?
It's getting energy, like
Well, using up energy, right? And using up energy to do what?
to fix things.
To fix? What else?
Connecting.
To what?
Connecting.
Connecting? Okay, that's another thing that it could be doing. So what are all those things? Fixing things, connecting, producing things. What is all that? Various what?
Function.
Functions. Exactly. So when the cell is not divided, it's just doing its own thing. It's doing its own function, specific functions. Right? So here is an example of a cell that's divided. But if a cell is not divided, then what is it going to be doing? Just sitting there doing nothing? No, it'll be doing its own thing. Doing its own functions, whether it's producing something, protecting something, fixing something, connecting something, communicating, whatever its function is specifically. Example, a bone cell. If a bone cell is not divided, then what is it doing? It's probably collecting minerals, gathering minerals to make bones strong. If a cell is, for instance, a cell of your of a gland cell, then what does it do? It's probably producing some type of secretion or product and secreting the product. Okay. So therefore, during interphase, cells are not divided, but doing its own function, specific to the cell, the nature of the cell, bone cell, muscle cell, epithelial cell, glandular cell, etc. Now, there is though a period during interphase. There is some moment during interphase before mitosis where the cell will make some preparations as well, prepare for division. Now, one of you said growth. Yes, the cell will be growing and maturing, but there's a moment when the cell will also, at some point, say, you know what? I'm going to divide. I'm going to divide. So to divide, the cell may make some preparations for division. So what are the few things that the cell can do during interphase to prepare for division? One thing that it could do is that the centrioles can replicate. The centrioles. Now remember, every cell has a pair of centrioles, right? A pair of centrioles. Every cell has a pair of centrioles. Those centrioles will replicate. Now you'll have two pairs of centrioles. Okay? So that's one thing it can do to prepare right before division starts. It'll multiply or replicate its centrioles. Another important thing that it can do is that the DNA that's in the chromosomes, or right, the DNA in the chromosomes, the DNA that's in the nucleus, the DNA will also replicate. So centrioles and the DNA in the nucleus replicate. In other words, like make copies of itself. Pretending that this is DNA, all these instructions and blueprints that I have here is DNA. I could put it through a copy machine and make an extra copy. Now I have two copies of that. That's what the chromosomes will, what the cells will do. Will prepare for division so that then we're ready to start out the process of mitosis. Okay, so interphase, you can put Roman numeral 1 there if you like, is the cell's not divided yet, but it can prepare for division, and then throughout most of its lifetime, it'll also be doing its own thing. Now, we can move on to number 2, Roman numeral 2. Again, you could use Arabic numerals or anything else, just list these things, but...
When you speak about time, like the time frame, that's important about interphase?
No.
Like what is that?
No, it's not important. The time frame can vary from one cell to another. The time frame of interphase of a cell that doesn't live too long could just be a few days. Whereas the time frame of an interphase of a cell that lives very long could be years or years.
Okay.
So, yeah. So the time frame doesn't matter. But yes, one thing that is different though is, with regards to time, is that interphase will be most of the cell's lifetime. What happens throughout most of the cell's lifetime. if this is the life cycle of a cell, probably this is when mitosis is occurring, and all the rest here is just interphase. Where there's growth, your classmate said growth, yeah, you'll see some letters like G for growth, G1, G2, asecond growth and maturation. You'll see S for when the cell is going to prepare for division. But as you can see from this lifetime circle here, a very small fraction of its time is just division. Most of the lifetime of the cell is, regardless of the cell, if it just lives for a few days or lives for years, it's still, interphase will be a major part of its life cycle. Okay, anyway, let me just erase that to not confuse you any further. But going back now to the next part, now the cell is prepared to divide. It has done these few things to prepare for division, centrioles and DNA replicated, but now we can move on to number 2, mitosis. The actual cell division of diploid cells. Again, the actual cell division of diploid cells. So let's look at the actual division of diploid cells. In fact, mitosis really refers to nuclear division. Nuclear division. Where this nucleus is actually going to be divided and directed cell division. So major thing here involved in cell division is not only the centrioles but also the nucleus. It's what drives, it's what will determine the cell division, the division of the nucleus. Okay? What about all the other things like the mitochondria and the cytoplasm and all the fibers that we have in the cell? That just follows afterwards. But what is important here is the nucleus determining and directing the cell division. So therefore, a lot of the events that we'll see are specifically to what's going on in the nucleus. Okay? So now let's take a look at mitosis specifically. And what you see with mitosis, you'll see in a moment, is that, by the way, these models help to explain mitosis. They're very simplified models used in biology class, so I don't really use these, but if you'd like to look at them later, you may. But you can see right here with the first one that we're ready to start out cell division. How do I know that? Because I see two pairs of centrioles. That means this is now ready to start cell division. Okay? Okay, in fact, this is already beginning cell division with the first phase of mitosis. So the phases of mitosis, the steps of mitosis are described as phases. The first one, number 1. Now you can use Arabic numerals if you like. The first phase of mitosis, or the first step, is called prophase. The word pro means the first, right? Or pre, right? The beginning. Now, many of you might start to wonder, how am I going to remember these phases in that specific order? It is important that you know the specific order of the phases of mitosis. And the first one is prophase. And that one's pretty easy. Most people can remember prophase as being first. Pre means first, or the initial one, right? Anyway, prophase is that first phase of mitosis. What happens during prophase? Well, now that the cell is ready to divide, this first step of cell division, called prophase, what will happen is that the nuclear envelope degenerates. The nuclear envelope, see how I erased the nuclear envelope? The nuclear envelope degenerates. So here, what happened to the nuclear envelope? It's pretty much dissolved. It's gone. It's gone. It disappeared. Okay? So there's no nuclear envelope now. The nuclear envelope or the nuclear membrane degenerates, breaks apart. Now, what else happens here during this period? The nucleolus is also gone. See the nucleolus? Well, I didn't have nucleolus there, but the nucleolus also is gone. Where'd it go? It just sort of dissolved, or you could say degenerated as well. So the nuclear membrane and the nucleolus
So how do you write this for prophase?
How do I draw that?
You want to write it, first to write about that,
About which?
which just happened, how everything fades away.
Oh, it just degenerates.
So just put
Degenerates. Yeah. The word degenerates is this. Degenerates, right? It means to... On sugar cubes. Ever put sugar cubes? Put it on a little tray and put hot water over it. What happens to the sugar
It
cube?
dissolves.
It's like gone, right? What happened to the sugar cube? It's gone. It's dissolved in the water, but that's what happens here. It kind of like dissolves or degenerates. It doesn't disappear forever. It's still there somewhere, but as you can see, I erased the nuclear membrane, and the nucleolus also degenerates. Okay. Now, is there anything else that happens here in this first phase? Yes, this first phase is really important, and that is because a couple more things happen. The chromatin. Now, remember that I had chromatin here. Those fine little threads representing chromatin. The chromatin, let me erase this word. I just put it there for the spelling. But the chromatin, which contains DNA, condenses to chromosomes. The chromatin, so here's your chromatin. Looks like fine little threads. But let me erase them, because now they will condense into what you're more familiar with. It looks like this. Like little wings of insects. Like little letters. Letter X's or Y's, whatever. Bunny ears, whatever. Okay? Here's an example of chromatin. Okay? Contained in DNA. But when it condenses into something that looks more like this. Something that perhaps you're more familiar with when you look at chromosomes. Looks like wings of a mosquito or some type of insect. So the chromatin, which contains DNA, is just going to condense now into a form that looks more like that. Condensed, right? Have you ever seen how they make pretzel sticks? Those places that they have in the malls called Annie's Pretzels or whatever. They have a lot of flour there. They put flour, they put some dough. It doesn't look like much at first. But then you see their hands, they're working. They're rolling it up into little pretzel sticks, and then they fold them into the typical shape of pretzels, right? Same thing with cigars. When people have cigars, just a lot of the stuff, right? The tobacco on the table. They take that, put it on the tobacco paper, and start rolling it up, condensing it into cigars, right? So that's pretty much what happens here as well. The chromatin condenses to chromosomes. So now we're going to have chromosomes present. Do I see that here with these models? I do see that. For instance, with this model, you can see some little, like colorful cotton-shaped things that look like little rods. Those are examples of chromosomes. So this is probably now part of prophase. This is also probably prophase because of what I see there. We have two pairs of centrioles. Now, moving on. The last thing that happens here, the paracentrioles. The paracentrioles that we had the two pairs of centrioles start to form these things that you see here. Let me erase the nuclear envelope. Start to form these things that you see there that look like the fine thread that a spider hangs from. They're called here, by the way, they call that spindle, the spider's spindle. And here that's what it's called also. It's called the mitotic spindle. So the centrioles, right, they start to move apart from each other and form those spindles, the mitotic spindle. Now you might wonder, what's the purpose of those spindles? There's no spider here. No, but we do have chromosomes. The chromosomes soon are going to attach to those spindles, like a spider attaches to its own spindle. Right? Anyway, those are a few things here that happen during prophase. Okay. questions about these many things that happen during prophase, this first early step? Okay, at the end, when we review all the steps of mitosis, I'll show you a little mnemonic or way to remember the steps in that specific order. Okay, so number 2. Now comes the second step of mitosis. Here, I believe it is probably this model. You can see some light blue little threads. Those light blue threads represent spindles. If not, that's okay. You can review this if you'd like later on, but that's not important because that will not be used on the exam. What will be, perhaps after the exam, is what happens during metaphase. That's the second step. Metaphase. So let me just fix this diagram a little so that we can look at metaphase. Okay, so now we no longer have a nuclear envelope, so the chromosomes are floating all around. But we do have 2 pairs of chromosomes, centrioles, another pair of centrioles. Okay, they form the mitotic spindles. Okay, and during metaphase, the chromosomes attach themselves to the spindles, and even more than that, the chromosomes line up along a central line like this. The chromosomes line themselves up along a central line that we call the equator. So the chromosomes attach to the spindles, and then the chromosomes line up along the equator. Line up or align themselves. You could say align as well. Align means to form like a sort of straight line. Align on the equator. Equator. How many of you have studied geography before, taken a geography course? Or looked at maps? I'm sure everybody's looked at a map. Which is the equator on the map?
The middle.
That middle line that you see. Now the middle line, it doesn't matter. It could be vertical, it could be horizontal. The cell is always moving. It's not going to always be in an upward position like that. The cell will be floating around. Could be upside down, could be pointed to the right point. It doesn't matter. It's a 3-dimensional structure, spherical structure, right? So what you do see though is that you see some imaginary, you see here an imaginary straight line. Whether it's like this, vertical or horizontal, that imaginary straight line that you see there formed by the chromosomes lining up like this is called the equator. So when the chromosomes start to line up in a straight line, align themselves along a straight line, it forms here a thing called the equator. When you see the chromosomes like that, you could then identify that as being metaphase. So that's what it does during metaphase. In fact, that's probably the only major thing that it does in metaphase. Okay, questions about metaphase? Okay, let's move on now to step 3, the next phase. The next one is called anaphase. The third step is called anaphase. What happens during anaphase? Well, you can see that I drew a chromosome here, example of a chromosome. You can also appreciate that in the middle of the chromosome, there's like a button-shaped structure where all the parts of the chromosome attached to in the middle, like a button on the middle of a bow tie, right? Anyway, the chromosomes then split at its center there, called the centromere. That little center that you see there called the centromere of the chromosome. So the chromosome split at its centromere. Let me erase this here. Right? So here's one half of the chromosome. The centromere now split in half as well. And the other half of the chromosome see, each half of the chromosome splits apart. These chromosomes will split at its center there, called the centromere. So now, if this was anaphase, now it would look more like this. Okay, same thing with the other side. Okay, see how they split apart? Okay? So that would be one thing here, that as they split apart, they start moving towards opposite sides. The chromosomes split at the center there, at the centromere, and then start moving towards opposite sides. Towards opposite poles, right? Half moving towards the right side, and the other half moving towards the left side. Okay? Or it could be half moving up and the other half moving down, depending on how the cell's going to divide. If you're going to divide from left to right or from up and down, whatever, it doesn't matter. Do I have anaphase here represented? So this was metaphase, as you can see the chromosomes sort of lined up in the middle there. But now, in anaphase, you can see how the yellow half of that yellow chromosome is going towards one side, the yellow half of the other, that same chromosome is going toward the other side. The same thing with the other chromosomes, the green one and the red one, or orange, they have split just like I drew here. So now they start moving apart from each other as well. The chromosomes split in halves and move to opposite sides or opposite poles. You could say poles as well. Okay? Also, one other thing that happens here during anaphase is that you see that the cytoplasm starts to slowly do this as well. The cytoplasm starts to also split. The process of the cytoplasm splitting is called cytokinesis. Cytokinesis begins. It begins as early as now during anaphase. What is cytokinesis? The cytoplasm also splitting. Okay? So as you can see, mitosis really refers to the many events happening within the nucleus and the chromosomes, but the cytoplasm will also many times divide as well. However, the cytoplasm dividing or cytokinesis is not actually mitosis. It is a process though, so that we do have two individual separate cells at the end. But we will have many times cells that will not divide like this by splitting the cytoplasm, and you'll end up with a cell that has what? If there's no cytokinesis, you would end up with a cell that has two nuclei. That is possible. There are many cells, for instance, some heart cells have two nuclei. Liver cells, many of them will have two nuclei. Okay. Most cells have, yes, one nucleus, but you will have cells where cytokinesis did not occur, and therefore you'll have a cell that's perfectly normal and healthy that will have two nuclei. But in order for most of our cells to have its own separate cytoplasm and nucleus, right? We want to also divide the cytoplasm, split the cytoplasm. Like you taking a big piece of dough and splitting it into two small pieces of dough so that you can make pizza one day and then pizza another day. Any questions up to there? So yes, this early step of cytokinesis can begin as early as now during anaphase. However, it's in the last step, number 4, telophase, So here, let me just erase this for a quick moment. During telophase, where the cytokinesis, right, will actually be completed, if it's going to take place. If there's going to be cytokinesis, splitting of the cytoplasm, it will complete during this last phase. Cytokinesis completed. So if I continue like this, that, by the way, this way, by the way, what I'm drawing here, this is called the cleavage furrow. Cleavage furrow. Okay. This cleavage means to cut. Cleave, to cut. A meat cleaver is for cutting, to split, right? So that's the cleavage furrow, to allow for the two cells to actually separate completely from each other. And then, what other things happen? The nuclear envelope reappears. In other words, the nuclear envelope is reassembled. Same thing here. The nuclear envelope again. Okay? Each one is going to have its own nucleus with a nuclear envelope. The nucleolus also is reestablished. Nucleolus again, you can also see a nucleolus on each one of the nuclei, within the nuclei. Right? And what happens to the chromosomes? The chromosomes go back again to being chromatin. Here's the chromosomes. These chromosomes, once again, become chromatin. So here, in the nucleus of each of these, you'll see chromatin. No longer chromosomes. The chromosomes are only visible during cell division. So by this phase, chromosomes, once again,
Loose again.
kind of like loose into flowers, right? Into, in this case, into just fine little threads called chromatin, right? So this now goes from chromosomes to chromatin. Okay? And now, each new cell, each new daughter cell, as you'll find, you'll have two identical daughter cells with the same number of chromosomes as the mother cell had, right? 46 chromosomes. 46 chromosomes. Meaning that these will also be diploid cells. Two, by the way, the number 2n is a symbol to represent diploid cell. 2n. Okay, those are the steps there of mitosis. Okay? So here, we finished completely like this. We will have two identical cells, right? That are a result of mitosis completed. Okay, this is the steps here of mitosis. Now, how to remember that order? If you could memorize the order: prophase, metaphase, anaphase, telophase. But one way that you can remember is you take the first letter of each word, and it spells out the word PMAT. PMAT, which you might use for a puppy mat, or for a puppy to do pee. Or I've seen other students use a little mnemonic, or some way to remember by making up a small story. Like, for instance, promise me anatomy on the telephone. If that helps you, great, use it. If not, make up your own little mnemonic, or use the PMAT, or just use your own memory. Say, I can remember them, I don't need mnemonics. I could do prophase, metaphase, anaphase, telophase. Now, it's important that you know that order. Okay? How about interphase? Remember, interphase is not part of mitosis. But after we're done, these two cells now will enter into its own interphase, period, when they do its own functions and activities and so forth. So interphase is not part of mitosis, but it is a large, important part of a cell's lifetime. But the main phases of mitosis are these 4 phases. 1 to 4, prophase, metaphase, anaphase, telophase. And that is also going to be helpful. If you learn this well now, it will make it easier for you in 204. Because those 4 phases are used once again in other types of cell division that you will discover, like in SCB 204, where you talk about reproductive cells dividing. And they will also go through those same 4 phases in that order. So learn it now well. When you get to 204, you say, I know all the phases in order. Good. And maybe a professor will make it, give you a smile, say, hey, this student knows well. Anyway, you'll impress the professor. Anyway, questions about mitosis? Questions? So let me erase here. Okay? And what you can do now is, why don't you take a microscope? So everybody grab a microscope. Okay? And then we have slides here on these trays. 2 different slides. One comes from a plant, I think it's onion root tips, from an onion root. And the other comes from fish, white fish. Now the trachea, I'm putting that down here, we're not going to be using it. But the onion root tip, the root tip, and the white fish cells, you can look at those to explore, look at, and try to identify as many of the phases as you can, including interphase. You'll see many cells that are in interphase, but try to identify those different phases with the microscope slides. Again, we have 2 varieties of cells there, the root tips and the white fish cells. Okay. Let's do that for a little while, and then we'll take a break sometime soon. Something fell out of your microscope.
It's mine?
No,
Here? Yours.
Miguel, who's your classmate next to you? Fell out of your microscope. Oh, there we go. If it can't be fixed, you can put that one back and take it. Okay, I need the sign-in sheet. Who's got the sign-in sheet?
Oh, yeah, thank you.
That's okay. That's ground. No,
Oh, you have a pen?
don't use the slides that are on the table yet. Those we'll use later on.
I'll take a pen.
Who's got the sign-in sheet?
Yeah, it's here.
Okay, guys, there's 2 different varieties of cells there, the onion root tip, the root tip cells, and the whitefish cells.